Today, the global biogeochemical cycles are driven by the cycling of two main elements. One is carbon, what life is essentially based around, and the other is oxygen. Primary productivity is carried out by the photooxidation of water, essentially oxygen in its reduced form, to reduce carbon dioxide to organic carbon. Conversely, energy metabolism is largely driven by oxidizing organic carbon to carbon dioxide using oxygen as the terminal electron acceptor, producing water. But how did these metabolic systems come about? Well, if you want to know how photosynthesis and respiration came into being in the first place, you can see my video, The Origin of Photosynthesis and Respiration. But the early forms of these metabolic systems didn't reduce or photooxidize oxygen. Rather, they used other elements like sulfur and iron. In this video, we'll discuss how the oxygen-utilizing or aerobic versions of these systems came into being. We find definitive evidence of large-scale oxygen production in the Neo-Archean, which spanned from 2.8 to 2.5 billion years ago. But some publications have suggested that there was limited oxygen production during the Mesoarchean, which spanned from 3.2 to 2.8 billion years ago. The evidence for this is that iron and manganese-based minerals from this time are partially oxidized, seemingly indicating that there was some oxygen production. However, a possibility that's all too often ignored is that instead of producing oxygen which oxidized the metals, ancient phototrophic organisms were photooxidizing iron and manganese themselves to reduce carbon dioxide to drive primary productivity. Now, I just said that most photosynthetic organisms on Earth photooxidize water, but this isn't technically true. What they do is photooxidize manganese in a manganese-oxygen-calcium cluster from the oxidation state 3 to 4, and the electron goes on to reduce carbon. The manganese-3 is regenerated when the manganese-4 in the cluster, also known as the oxygen-evolving complex, oxidizes a water, producing molecular oxygen. This would suggest that oxygenic photosynthesis itself was preceded by a process which photooxidized manganese. Furthermore, the oxygen-evolving complex is produced by photooxidizing the soluble manganese-2, further reinforcing this hypothesis. The oxygen-evolving complex is sandwiched between the D1 and D2 subunits within the photosystem II complex, and these are homologous, i.e., they evolved from a single ancestral subunit, which we can call D0. In a metal-oxidizing phototroph, this could have been the site where either iron 2 was oxidized to hematite or manganese 2 was oxidized to bixbyite. Both of these reactions would require 2 photons, one per each metal atom, but in the case of manganese, a second photon could oxidize one of the manganese from manganese 3 to 4, producing sodium manganese oxide and thus providing another electron to reduce carbon with. After D0 was duplicated, the resulting subunits could produce 2 sodium manganese oxides between them. Now, they're the same number of manganese atoms in the same redox states as the oxygen-evolving complex. At this point, a calcium ion could replace the 2 sodium ions, forming a cluster. Now, all the manganese atoms can be oxidized by 2 more photons, all the way to manganese 4, giving the organism 2 electrons for every manganese metabolized to reduce carbon with. But, to milk the manganese for all it's worth, the organism can oxidize one of the manganese atoms in the cluster to manganese dioxide, getting 1 more electron and partially oxidizing water. At this point, the photosystem could either release the cluster or bind a manganese 2 from the environment, which would have reduced one of the manganese 4s in the cluster back to manganese 3. Both the original and new manganese oxide would be photooxidized to manganese 4, and the new one would be again oxidized further to manganese dioxide. A third exogenous manganese 2 will enter the cluster, and the cycle repeats. At this point, some of the manganese atoms in the cluster could be bound to the amino acid residues within the photosystem complex, effectively making it an enzyme cofactor to oxidize more manganese ions. However, because the redox potential of manganese 3 to 4 is greater than that of water to oxygen, it could begin oxidizing water. Initially, this can only be done in limited quantities because without being well coordinated by the enzyme amino acid residues, this reaction would likely produce hydrogen peroxide as a byproduct, a highly toxic chemical. Once the residues could coordinate it, the complex would start producing diatomic oxygen, giving rise to oxygenic photosynthesis. But when we compare oxygenic and anoxygenic phototrophs, a number of differences between the ways they use substrates are present. For one, oxygenic phototrophs have two varieties of photosystems in their light harvesting machinery, while anoxygenic phototrophs will only have one per operational taxonomic unit. It's believed that at some point, all of these evolved from an ancestral photosystem that then split into type 1 and type 2 photosystems. The type 2 would give rise to the photosystem 2 complex of oxygenic phototrophs, which contains the oxygen evolving complex, along with that of the chloroflexi and purple bacteria. Meanwhile, the type 1 evolved into the photosystem 1 complex in oxygenic phototrophs and the photosystem of acidobacteria, chlorobi, and heliobacteriaceae. If you look at the electron transfer sequences, you can see the similarities between photosystem 2 and the purple bacterial photosystem and photosystem 1 and the green sulfur bacterial photosystem. Although, after the progenitor of the first oxygenic phototrophs acquired both types of photosystems, various aspects of the type 1 system were incorporated into the type 2, such as the PSA and PSB subunits. Another feature unique to oxygenic phototrophs is a thylakoid membrane, which houses all of the light harvesting molecular machinery. Anoxygenic phototrophs have their photosystems embedded within their plasma membrane. While it was traditionally believed that thylakoids evolved from vesicles which branched off from the plasma membrane, the fact that they form from dissolved lipids which arrange into bilayer membranes in the cytoplasm indicate that this is unlikely. While most cyanobacteria have thylakoids, one group, the Gliobacter clade, lack them, instead having their photosystems in their plasma membrane. Molecular clock estimates have indicated that Gliobacter split off from the rest of cyanobacteria 2.5 billion years ago. While we have evidence of oxygen production throughout the Neoarchean, it's not until the start of the Proterozoic 2.5 billion years ago that we see oxygen building up in the atmosphere. One thing to note is that if all life on Earth suddenly vanished, within 1 million years, all of the atmospheric oxygen would be gone, having oxidized reduced minerals in the rocks. So, while oxygen was being produced by phototrophs in the Neoarchean, it wasn't until the Paleoproterozoic that it was produced faster than oxidative weathering could remove it from the atmosphere. This means that the evolution of the thylakoid membrane allowed oxygen to be produced fast enough for atmospheric buildup. Within the thylakoid membrane, electrons taken off water molecules are passed from photosystem 2 to a membrane plastoquinone molecule. This is a comparatively less electronegative molecule, and it's transferred to the more electronegative plastocyanin via a cytochrome BC oxidase. Cytochromes are proteins which contain a porphyrin with an iron atom in the center, which facilitates the flow of electrons through complexes. Since the electron is going from a less to more electronegative molecule, the reaction is exothermic, i.e. energy producing. The cytochrome BC oxidase uses this to pump protons from a lower to higher concentration, and the resulting cross-membrane gradient is used to drive the ATP synthase enzyme. The plastocyanin is then oxidized in the photosystem 1 complex, and the electron is then transferred to a ferredoxin. Finally, the ferredoxin NADP reductase oxidizes the ferredoxin to reduce an NADP to NADPH, where it's then used to reduce carbon to be assimilated. Plastoquinone is a simplified version of menaquinone, a membrane-bound electron shuttle used by both anaerobic respirers and anoxygenic phototrophs, which itself is a simplified version of methanophenazine, a carrier used by methanogens. So, it probably evolved from a menaquinone used by an ancient photothiotrope. The cytochrome BC complex used by oxygenic phototrophs would have come about by adding an additional anchoring helical peptide, which became its subunit 4, to a B-type cytochrome. One purported function of cytochromes in anaerobic respirators is to chelate the poorly soluble iron 3 so it can be held in an enzyme. In dissimilatory iron reducers, the iron would act as the electron acceptor in the oxidation of carbon. The progenitor of the cytochrome oxidase would have been a surface enzyme, which passed the electron from some kind of quinone to ferric iron, using the energy to pump protons. Finally, a cytochrome, which was bound to a risque protein, was added to it, which became the F subunit. As for NADPH, it's a modified version of NADH with an extra phosphate group, and NADH has been used as an electron carrier since at least the time of Lucca. Oxygenic phototrophs use the Calvin-Benson cycle to fix carbon. The carbon dioxide is made part of an organic molecule when rubisco binds it to a 5-carbon ribulose biphosphate, which converts it into a short-lived 6-carbon intermediate, which then breaks down into 2,3-phosphoglyceric acid molecules. These are then reduced to glyceraldehyde 3-phosphate via NADPH and ATP. 2 of these are then combined to make glucose. The ones that aren't are combined into ribulose biphosphate for the next turn of the cycle. Enzymes which resemble rubisco, known as rubisco-like proteins or RLPs, have been observed in various prokaryotes. It's uncertain as to what lineage they arose in, but it was likely an archaeal methanogen. Most of these carry out utilization reactions involving the transfer of thiol groups from methylthioadenosine to amino acids to form methionine. Since the substrate of its progenitor would be a ribose, i.e., the sugar part of the methylthioadenosine, the addition of other amino acid residues at the active site would allow the original rubisco enzyme to add a carbon dioxide to the enol group, resulting in the formation of a carboxyl group. As for the Calvin cycle itself, not much research into its evolutionary history has been done. However, most other organisms which use it are aerobic lithotrophs, or organisms which power their energy metabolism via oxidizing inorganic molecules using oxygen as the electron acceptor. Now, purple sulfur bacteria use the Calvin cycle, but these evolved long after oxygenic photosynthesis began. Lastly, oxygenic phototrophs use porphyrin-based molecules with the magnesium in the center known as chlorophylls to harvest light, and these are a simplified version of the bacterial chlorophylls used by anoxygenic phototrophs. In a manner similar to the menaquinones in methanophenazine, these are simplified versions of respiratory hemes, with the iron swapped out with the magnesium. Once oxygen was being produced, it could act as an electron acceptor by respiratory organisms who would have been using other electron acceptors like the oxidized forms of sulfur and iron. In modern aerobic heterotrophs, NADH and FADH2, which were formed via the oxidation of other organic molecules, are oxidized by the respiratory complex 1 and 2 respectively. The respiratory complex 1 pumps protons against a concentration gradient, and both complexes 1 and 2 pass the electrons to membrane carriers known as ubiquinones. These are then oxidized using cytochrome C as the electron acceptor, and this reaction is facilitated by the respiratory complex 3, which again uses the flow of electrons from a less electronegative donor to a more electronegative acceptor to pump protons against a concentration gradient. The cytochrome C is then oxidized by the respiratory complex 4 using oxygen as the electron acceptor, and the energy of the reaction is again used to pump protons into the intermembrane space. This cross-membrane gradient is also used to drive the ATP synthase enzyme. Phylogenetic analysis of these complexes indicate that the system came out as an amalgamation of components of other microbial metabolisms. The evolutionary history of the respiratory complex 1 dates back to the days of the last universal common ancestor of all life. Back then, early organisms possessed hydrogenase enzymes, which oxidize hydrogen to reduce NAD to NADH to be used to reduce carbon. Acetogens still do this at the present day. Shortly after, methanogens evolved hydrogenases with modules to pass the electrons to membrane-bound carriers such as methanophenazine. In the first respiratory organisms, which use sulfate as the terminal electron acceptor, one of these hydrogenase complexes acquired both the NADH and membrane carrier binding sites. At this point, it would no longer be able to oxidize hydrogen, and instead pass electrons from NADH to a cytoplasmic electron carrier, although this carrier would have been menaquinones versus methanophenazine, and the complex would have developed the ability to pump protons. The menaquinones would have then shuttled the electrons to the APR and DSR enzymes, which reduce sulfate to sulfide. Later, the addition of iron-sulfur clusters on more amino acid residues at the N-terminus of the membrane carrier binding module would allow the complex to reduce the high-potential ubiquinones versus the low-potential menaquinones. Because the difference in redox potential between NADH and ubiquinones is greater than the difference between it and menaquinones, the later could pump more protons per NADH metabolized, 4 in total in aerobic organisms. The complex 2 would have originated from a fumarate reductase enzyme. While only aerobic respirers use the TCA cycle as a means of energy production, anaerobic organisms use the horseshoe TCA cycle, where the right half runs oxidatively and the left half runs reductively, with the oxidized and reduced forms of NADH being cycled between the two branches. This version is used to synthesize alpha-keto acids, which are then converted into amino acids. The fumarate reduction step was reversed, and FAD, which nominally exists in enzymes as a cofactor versus floating around in the cytoplasm like NAD, acted as the electron acceptor. Complex 3 is a B-type cytochrome oxidase, which is homologous to the cytochrome oxidase used by oxygenic phototrophs, known as cytochrome B6F, with 2 subunits merged into one. Instead of oxidizing and reducing plastoquinone and plastocyanin, the ancestral complex 3 began oxidizing and reducing ubiquinone and cytochrome C. Complex 4 is unique in that it possesses several copper cofactors in both the intra-enzyme electron transport chain and at the active site where oxygen is reduced to water. This complex likely evolved from a nitric oxide reductase enzyme, or NOR, which reduces nitric oxide to nitrous oxide. Nitric oxide reductase is used in the denitrification pathway, which uses oxidized nitrogen species as the electron acceptor to oxidize carbon. But oxidized nitrogen species wouldn't have been common before the advent of molecular oxygen. However, many non-denitrifying organisms possess NOR to detoxify nitric oxide, and on the early Earth, photochemical reactions involving UV light would produce nitric oxide. So, ancient microbes likely also used nitric oxide reductase to detoxify it. Again, substrate similarities allowed it to metabolize the newly produced diatomic oxygen. If I were to guess, the immediate progenitor of the aerobic electron transport chain was one used by an iron respirer. In a manner similar to modern aerobic organisms, the protocomplex 1 would oxidize NADH and pass the electrons to ubiquinones, which would then go to a protocomplex 3, which was ancestral to both the B6F used by oxygenic phototrophs and the BC1 used by modern aerobic organisms. The electrons would then be passed to a ferric iron ion chelated by cytochrome C, which would act as the terminal electron acceptor. The organism would also use cytochrome C to obtain electrons from ferrous iron to pass them to a protocomplex 4, which used the electrons to detoxify nitric oxide. When these two systems were bridged by cytochrome C, the nitric oxide reductase would then start reducing oxygen, which then replaced iron as the terminal electron acceptor, giving rise to the first aerobic respiratory system. As stated before, anaerobic respirers lack an oxidative TCA cycle and instead have various enzymes strip electrons from different organic molecules and pass them to NADH. As such, individual OTUs of anaerobic respirers are incapable of completely oxidizing organic carbon. Once the fumarate reductase enzyme had been reversed to become the succinate dehydrogenase, the two branches of the TCA cycle could be connected, and other upstream elements of the left branch were reversed, completing the cycle, while the production of more NADH would provide additional electrons to be fed into the respiratory electron transport chain. Since acetyl-CoA is a near universal part of all metabolic pathways, including the TCA cycle, the advent of the oxidative TCA cycle allowed early aerobic organisms to completely oxidize organic carbon to carbon dioxide. Now, I should mention that there are reductive versions of the TCA cycle used by some anaerobic organisms like green sulfur bacteria to fix carbon, and this has been argued by many to be the carbon fixation pathway used by the first organisms. However, phylogenetic analysis of the reverse TCA cycle enzymes don't indicate it predates LUCA, and the anaerobic taxa which utilize it evolved after the first aerobic organisms appear. This suggests that the horseshoe TCA cycle was employed by the last common ancestor of everything alive today, which then gave rise to the oxidative TCA cycle, and then the reductive TCA cycle came out as a reversal of it. Once the ability to utilize oxygen evolved, ancient microbes could use it to oxidize various inorganic compounds as well. Hydrogen would have already been oxidized using carbon dioxide, sulfate, and ferric iron as an electron acceptor, utilizing hydrogenase enzymes to take the electrons from the molecule. Instead of being passed to menaquinones, they'd be passed to ubiquinone and sent down the aerobic electron transport chain, giving rise to aerobic hydrogenotrophy. To fix carbon, these organisms would have obtained the Calvin cycle enzymes from early oxygenic phototrophs and passed some of the electrons from the hydrogenase enzyme to an NADPH, where they would then go on to reduce Calvin cycle intermediates. So, aerobic hydrogenotrophy can be seen as a hybrid metabolism, having obtained components from aerobic respirers and oxygenic phototrophs. Organisms were likely oxidizing both sulfur and iron prior to the advent of atmospheric oxygen, but they would have been doing so photochemically to reduce carbon dioxide. In the case of sulfur, they would have been using the reversal of the sulfate reduction pathway. But once oxygen was present, they could use it as an electron acceptor to oxidize sulfides to sulfate, producing energy. Initially, chemotrophic sulfur oxidizers would have used the reverse sulfate reduction pathway enzymes, DSR and APR, to strip electrons off sulfide. From there, the electrons could be transferred to the complex 4 of the electron transport chain, where they'd reduce diatomic oxygen to water. While some chemotrophic sulfur oxidizers use the reverse sulfate reduction pathway, more frequently they'll utilize one of two pathways: the SQR or the SOX pathway. The SQR, or sulfide quinone reductase, oxidizes sulfides to produce polysulfides, and then uses the electrons to reduce quinones to then be fed down the electron transport chain. These enzymes likely evolved from the respiratory complex 1. The polysulfide is then oxidized to sulfite via rhodinase enzyme. These enzymes are commonly used by other organisms to detoxify cyanide by reacting them with thiosulfate, producing thiocyanate. From there, either the reverse sulfate reduction enzymes, SAT and APR, will oxidize it to sulfate, or a sulfite dehydrogenase will, and one subunit of this would have evolved from a cytochrome, and the others from a molybdopterin oxidoreductase. The enzymes involved in the other sulfur oxidation pathway, i.e. SOX, evolved from cytochrome oxidases, where the cytochrome oxidizes sulfur and passes the electrons to a dioxygen reductase. This pathway begins with SOX-XA binding a sulfide to SOX-YZ via a disulfide group. SOX-CD will then incrementally oxidize it to a thiosulfate group, which is finally oxidized to sulfate by SOX-B. Phylogenetic analysis has indicated that the first enzyme in this complex to evolve was SOX-CD, which dates to roughly 3 billion years ago. This would have been slightly earlier than the advent of oxygenic phototrophy, so it may be that it was originally used by photothiotrophs, and it would have evolved from a molybdopterin oxidoreductase that was bound to a cytochrome. While various sulfur oxidizers use the reverse sulfate reduction pathway, this one can only oxidize sulfide to sulfate. They cannot oxidize other partially oxidized forms of sulfur like thiosulfate and tetrathionate, so they likely originally used the ancestral SOX-CD to metabolize these compounds. SOX-CD is homologous with the sulfite dehydrogenase, used by some prokaryotes which employ the SQR pathway. So, one likely branched off from the ancestral SOX pathway and was incorporated into the other pathway via horizontal gene transfer. The next one to appear was SOX-B, which came about around the same time oxygenic photosynthesis began 2.8 billion years ago. This enzyme would have negated the need for the APR and SAT enzymes to oxidize sulfite to sulfate, decoupling the SOX pathway from the reverse sulfate reduction pathway. SOX-B was originally a 5' nucleotidase, which would have used its prior phosphate esterase mechanism to cleave a thioester bond. So, it may be that it originally acted as a substitute for SAT. About 2.6 billion years ago, we see SOX-XA and SOX-YZ emerge. The formation of the disulfide bond from an exogenous sulfide would put it in an intermediate sulfur redox state, which SOX-D had evolved to metabolize. The involvement of these two enzymes then allowed sulfide to be fed into the SOX pathway, and along with the advent of SOX-B, sulfur could now be converted from the most reduced to most oxidized form, maximizing the energy obtained per sulfur atom metabolized. But organisms have to do more than obtain energy. They also need building materials to construct biomass out of. While the first chemotrophic sulfur oxidizers likely obtain carbon in the form of organic matter, modern sulfur oxidizers can also get carbon from carbon dioxide. Now, you might ask, how is this possible, since sulfur is more electronegative than carbon? Well, the respiratory complex 1 nominally oxidizes NADH to reduce ubiquinones. So, to use electrons from sulfide to reduce carbon, the electrons are first taken from sulfide by an SQR, where it reduces a quinone. From there, it's taken to a respiratory complex 1 that functions in reverse. The flow of protons from a high to low concentration is used to move an electron from a more electronegative donor to a less electronegative acceptor. The NADH is then fed into the Calvin cycle. So, it was the reversal of a respiratory complex and acquiring the Calvin cycle genes from an oxygenic phototroph, which allowed sulfur oxidizing chemolithotrophs to transition from heterotrophy to autotrophy. The evolution of chemotrophic iron oxidation also resulted in the convergent evolution of different pathways. A common theme is that pretty much all of them take an electron from a ferrous iron and use it to reduce a C-type cytochrome, but different enzymes facilitate this redox reaction. Some use the iron oxidase genes, or IRO, iron-sulfur proteins with a cluster that has a much greater redox potential than ferredoxins, which allows them to oxidize ferrous iron. Others use the FOX genes, which likely evolved from a cytochrome, which was duplicated into two subunits, with one evolving enzymatic function. Still others use a sulfocyanin, a protein with a serine-like structure, with the iron swapped out with a copper atom. The cytochromes are again oxidized by a homologue of the respiratory complex 4 using oxygen as the terminal electron acceptor. So, unlike sulfur oxidation, where the enzymes used to reduce it were reversed, chemotrophic iron oxidation began with novel enzymes. With the advent of diatomic oxygen, another element that could be oxidized as a means to produce energy is nitrogen. When organic matter is remineralized, the nitrogen bound in it is released as ammonium. This is oxidized for energy in a process known as nitrification. Here, ammonium is first oxidized to hydroxylamine, then nitric oxide, next nitrite, and finally nitrate. The intermediate electron acceptors for ammonium oxidation are ubiquinones, but for the latter intermediates, it's C-type cytochromes, and, as in aerobic heterotrophs, a cytochrome BC oxidase transfers the electrons from the ubiquinone to the cytochrome, again pumping protons. The enzyme which catalyzes the first step is ammonium monooxygenase, and this would have evolved from a methane monooxygenase, which would again come down to the fact that the reactions which they catalyze are quite similar. In fact, modern ammonium monooxygenase enzymes can hydroxylate methane and other small alkanes. The next step, the conversion of hydroxylamine to nitric oxide is carried out by the hydroxylamine oxidoreductase. For a while, the evolutionary history of this enzyme remained elusive, but phylogenetic analysis revealed that they had evolved from a cytochrome C nitrite reductase used in another pathway known as denitrification, which we will discuss shortly. These are close relatives of the enzymes which produce nitric oxide and diatomic oxygen, and hydroxylamine oxidoreductase seems to have come about via the additions of amino acid residues to the N-terminal. The nitric oxide is oxidized to nitrite by an unknown enzyme, but the final step of the reaction is catalyzed by the nitrite oxidoreductase. This enzyme is part of the large melptoterrin oxidoreductase enzyme family, but how it fits into the family tree is uncertain. Like chemotrophic iron and sulfur oxidizers, nitrifiers use the reverse respiratory 1 complex to reduce NAD with electrons originally obtained from ammonia, and use the Calvin cycle to fix carbon. Now, onto the quote-unquote reverse of nitrification, denitrification. In lieu of oxygen, some prokaryotes can use oxidized nitrogen species as a terminal electron acceptor. In this pathway, first, nitrate is reduced to nitrite, followed by nitric oxide. This is then reduced to nitrous oxide, and finally, diatomic nitrogen. The first step is catalyzed by the nitrate reductase enzyme. These are also in the melptoterrin oxidoreductase enzyme family, but are only distantly related to the nitrite oxidoreductase used by nitrifiers, meaning the two evolved the ability to catalyze this reaction independently. Meanwhile, the enzymes which catalyze the next three steps, that being nitrite reductase, nitric oxide reductase, and nitrous oxide reductase, are heme copper oxidases, an enzyme family which includes the respiratory complex 4. The first enzyme in this family was likely a nitric oxide reductase, and the catalytic site probably came from a glutathione S transferase. These enzymes detoxify harmful exogenous compounds by binding them to a glutathione. As discussed before, the original nitric oxide reductase was likely for detoxifying nitric oxide versus energy metabolism. The incorporation of copper atoms as cofactors would allow it to reduce more electronegative compounds than other enzymes could. While a nitric oxide reductase was in all likelihood the progenitor of the dioxygen reductase used by aerobic respirators, exactly how this transition happened is a bit of a mystery. Although there are obvious substrate similarities between nitric oxide and diatomic oxygen, the reduction of the former to nitrous oxide takes 2 electrons, while the reduction of the latter to water requires 4. In the nitric oxide reductase, the electrons are transferred to the nitric oxide via a heme and a non-heme iron at the active site, where the iron atoms go back and forth between the oxidation states 3 and 2. In the respiratory complex 4, there is a single heme which goes between the states 2 and 4. A third electron is derived from a copper ion which goes between states 1 and 2. This leaves 1 electron unaccounted for. One hypothesis is that the 4th electron comes from a tyrosine at the active site. What's problematic about this for the evolution of oxygen reduction is that if you had a nitric oxide reductase reduce an oxygen with its 2 electrons, it would produce hydrogen peroxide, which is very toxic. i.e., if a nitric oxide reductase had evolved the ability to metabolize diatomic oxygen before it could transfer 4 electrons, it would be strongly selected against. However, since 2 nitric oxides are metabolized for every nitrous oxide produced, a total of 4 electrons are delivered to the active site. But since each molecule metabolized receives 2 electrons, the production of hydrogen peroxide would still be a problem for a nitric oxide reductase that metabolized molecular oxygen. One possible scenario is that early nitric oxide reductases were inadvertently reducing diatomic oxygen, producing hydrogen peroxide as a harmful byproduct, and the addition of components at the active site which delivered more electrons to each molecule was an adaptive response to reduce it all the way to water. After that, the respiratory complex 4 could act as the electron acceptor in the main energetic electron transport chain, at which point it would specialize in metabolizing oxygen versus nitric oxide. Modifications to the active site would also allow other nitric oxide reductases to metabolize the upstream and downstream steps of the denitrification pathway. The fact that nitrite reductase evolved before nitrification, which would have made denitrification a viable means to produce energy, suggests that it was also originally used for detoxification. Although not as reactive as nitric oxide, nitrite is still quite harmful to living cells. Once atmospheric oxygen was produced, volcanic lightning would produce nitrite along with a comparatively inert nitrate at an accelerated rate. So, nitrite reductase probably evolved as an adaptive mechanism to cope with this. Meanwhile, nitrous oxide reductase would have evolved as a way to get 2 more electrons, and thus more energy, from carbon oxidized. Because of the intertwined history of the enzymes used in aerobic respiration and denitrification, many aerobic heterotrophs can facultatively use denitrification if conditions become anoxic. Furthermore, many sulfur and iron oxidizers will use nitrate instead of oxygen as the terminal electron acceptor. In our cells, energy metabolism is carried out by mitochondria, and these were once free-living bacteria that were engulfed by the progenitor of eukaryotes. Like aerobic heterotrophs, our mitochondria have a respiratory complex 4, also known as a cytochrome oxidase, and among aerobic organisms, there are 3 main kinds of cytochrome oxidases. There's type A, which our mitochondria possess, type B, or the BA3 oxidases, and type C, or CBB3 type oxidases. Of these, it appears that type C were the first to branch off from the rest. Based on how its gene phylogeny compares with the 16S rDNA phylogeny of bacteria which possess it, it appears that the C-type cytochrome oxidase emerged in the Neoarchean. Furthermore, a subunit of the nitric oxide reductase closely resembles the monohemic subunit of the C-type cytochrome oxidase. Iron-oxidizing bacteria, which live in acidic environments, possess enzymes responsible for the synthesis of the porphyrin in the heme group, which resides in the cytochromes, which have features characteristic of the enzymes which produce the A and B type cytochromes. This suggests that the common ancestor of the A and B type cytochrome oxidases was an acidophilic iron-oxidizing bacteria, and the A type was later transferred to an aerobic heterotroph, which later became our mitochondria. For a long time, it had been known that the ancestor of mitochondria had been in the diverse alpha proteobacteria clade, but exactly how it fit into its family tree remained uncertain. Analysis of the COX gene cluster indicated that Iodidamonas was most likely the sister group of mitochondria. More specifically, the subunits are arranged in the same way in Iodidamonas and mitochondria. Not only have we identified the closest relatives of our mitochondria, we've also identified the closest relatives of our ancient ancestors who engulfed them billions of years ago, in a process known as endosymbiosis. We had long known that this cell was either an archaea or a close relative, since the molecular biology of eukaryotes is more similar to them than it is to bacteria. In deep-sea hydrothermal vents, we found a nucleate of prokaryotes similar to archaea known as the Asgard archaeota, who have a number of proteins such as GTPases, ESCRT, and SNF7, which they share with eukaryotes that they don't share with archaea, indicating that they are a sister group to the eukaryotes. Although it's not clear yet if they are a sister group or if eukaryota is nestled within them. Asgard archaeota are obligate anaerobes, getting much of their energy via fermentation. Now, one distinguishing feature of eukaryotes is the presence of membrane-bound organelles, such as the endoplasmic reticulum, the Golgi apparatus, and of course the cell nucleus. So, when discussing the origin of eukaryotes, one question that comes up is whether mitochondria were acquired before, after, or during the evolution of these organelles. There's still much debate that surrounds this question with no clear answer yet, but if I were to guess, I'd argue that mitochondria were acquired shortly after intracellular membrane-bound structures began to form. As the progenitor of the eukaryotes began to expand in size, the square cube law would mean that its surface area to volume ratio would decrease. In order to increase the surface area over which to absorb nutrients, invaginations of the membrane began to form. Shortly after, in order to consume particulate versus dissolved organic matter, some of these invaginations evolved the ability to split off into the cell completely once it came in contact with the food particle. The food particle would then be digested, and the resulting dissolved organic matter would be utilized by the cell. Today, this is how some unicellular eukaryotes such as amoeba feed. At some point, a cell engulfed a free-living aerobic bacteria, but instead of digesting it, the two formed a symbiotic relationship. Once this relationship was established, genes which coded for proteins which performed the same function as those in the host cell were deleted, because it could now get them from the host itself. Other genes were incorporated into the host's own genome, and this process is known as genomic streamlining. Many of the genes transferred from the nascent mitochondria to the nucleus were involved in metabolic processes, and this is why modern eukaryotes have a metabolism more similar to bacteria with similar lipid and carbohydrate compositions, while having a molecular biology more similar to archaea. As this happened, some of the invaginations would have split off completely, becoming primitive organelles. Over time, they would have become specialized for different functions, including act as the genome encapsulating membrane, i.e. the nucleus, at which point the cell became the first eukaryote. It's believed that the progenitor of mitochondria were engulfed 2.2 billion years ago, with the last common ancestor of eukaryotes living 2 billion years ago. It was during this time that oxygen began to build up in the atmosphere. Some eukaryotes also have a different type of organelle, i.e. chloroplasts, which carry out oxygenic photosynthesis. Like mitochondria, these were once free-living bacteria which were engulfed by a eukaryote, and molecular clocks indicate that this happened about 1.6 billion years ago, during the transition from the Paleoproterozoic to Mesoproterozoic. This prokaryote was a cyanobacteria, most closely related to the nitrogen-fixing Nostochales. In a manner similar to the mitochondria, many of the genes of the chloroplast were either deleted or moved into the nuclear genome, although chloroplasts retain a larger portion of their original genome than do mitochondria. The clade which is descended from the eukaryote which engulfed the ancestral chloroplast is known as the archaeplastids, and it encompasses most eukaryotic algae and land plants. The algae who aren't archaeplastids acquire their chloroplasts by engulfing archaeplastids in a process known as secondary endosymbiosis, and these include euglenids and brown algae. The inclusion of the ability to use and produce oxygen in eukaryotes would ultimately facilitate the evolution of complex multicellular life forms. So, overall, we can see dissimilatory oxygen metabolism as the culmination of climbing the redox ladder. Earlier forms of respiration and photosynthesis, those which initially used sulfur and later iron, laid the foundation for aerobic metabolism. By looking at these systems used by modern organisms, we can trace how oxygen-utilizing metabolism came about via the development of systems which could handle higher and higher energies and redox potentials. Some examples include the evolution of cofactors like chlorophylls from bacterial chlorophylls, who themselves came from hemes, and ubiquinones from menaquinones, which came from methanophenazine. From these, we can trace the origin of systems which make the biosphere as we understand it possible from metabolic processes used by the last common ancestor of all life. In the future, we'll look at how the evolution of these systems impacted and was impacted by the world around them and the ecology of the Archean Earth.